Welcome to Grumpy Science, again with Bob and Mark, the Grumpy Scientists, coming to you straight from Bob's lab, Secret Science Laboratory, here in Chapel Hill, North Carolina.
I thought this was a pretty nice studio, but you know that's sort of the way it goes. Bob tricked me into coming here. He said we have a whole studio set up for this. Well, this video we're going to talk about one of our measures and one of the key elements of one of those measures. And this is one of the measures that we started off studying many years ago. We started studying it actually in animal studies a long time ago, back in the 1980s. It tells you how old I am. But a guy named George von Bekesche in the 1950s and 60s discovered just how the brain worked in terms of how different cortical ensembles work together. And he was doing this just through sensory perception. In other words, he was just using the sense of touch to figure out how things in the brain interacted. And everybody kind of said, yeah, he's kind of full of it until he won the Nobel Prize. But still, he did pretty well. And we replicated a lot of his predictions. We did a lot of experiments that were guided by his predictions. And basically,
doing some high-tech multimedia here.
Basically, here's our fancy PowerPoint.
Oops, gotta put my bottle opener back.
And what that meant was that if you had input from the skin, it goes up to the brain. So here's the skin down here. Goes all the way up to the spinal cord, goes into the brain, and it goes into a column of cells. These columns of cells, actually, when they get activated, that's what this little plus sign means, they're all getting activated. What they do is they start inhibiting the cells next to them.
So these are cells in the cortex, and this is a little plug of cells going down into the cortex, your brain.
Right. So every time you get an input to the skin or an input anywhere, you get activation, which is a plus sign, and inhibition, which is a negative sign. So basically what's going on, and this happens everywhere at all different levels. And if you're familiar with the term fractal, it's a fractal organization. So that tiny little cell organization, tiny little cell columns or mini columns inhibit their neighboring mini columns, and then those mini columns aggregate into columns. Those columns aggregate into larger groups, and those groups aggregate into larger areas, but they all have this one principle where they interact with one another. And the best way to think about this, well, there's a couple ways to think about it. If you're old as us, you can remember when you had an old television set and it had a contrast knob on it. And if you put it, and when you turn it on and you turn that contrast knob, some things got sharper. And what this inhibitory signal does is makes things sharper. And it's a little bit like looking at a big crowd of people, and the people that are just a little bit bigger would put their hands on the shoulders of the people next to them and push themselves up. And all of a sudden, the bigger people or the stronger people get a little bit higher because they want to be heard. And so it's basically all about fighting between different cells in the brain. Well, how would you measure that? And basically, why would you measure it? And the reason you measure it is because when things go wrong, this doesn't work anymore. And that's one of the things that we wanted to study, was that basically, I need to put my beer down, I don't want to spill it.
That would be bad.
Basically, when, we do have rules here. One is don't spill the beer. But whenever this column gets activated, its nearest neighbors gets inactivated. And so it's basically a push and pull system. And when things are wrong, don't work well, that push and pull system gets modified. But we wanted to figure out, how do you measure it? And it turns out there's very few ways to measure it. And really the only really good way to measure this, and easiest way, is with tactile perception, like you do with a brain gauge. So what we do is we actually deliver two inputs. And so we activate two places in the brain that are side by side.
Fingertips.
And
side
basically,
by side on your
you activate
cortex.
two places in the brain that are side by side. The place in the middle goes negative, And you can tell now you can tell the difference between those two. And then you get a fairly decent score on your amplitude discrimination, because we deliver like one stimulus to this that's actually, say if you measured it, it would be up here. Another stimulus that would be a little lower. And you can tell the difference between a large stimulus and a smaller stimulus. But when things go wrong, you can no longer tell the difference. How's that for a simple explanation? So how do we do this with a brain gauge? And how do we actually measure that and take into account all the differences from one person to another? Because here at grumpyscience.com, the first thing we say is, why does everybody grouch about this? Yeah, you got to worry about, thickness of the skin and all this kind of stuff. And we had one guy tell us, oh, you can't do this because some people's hands will be cold, which is totally ridiculous. But anyway, what we do is we do what's called the amplitude discrimination test. We deliver one stimulus here, and then we deliver another stimulus here, and we see if you can tell the difference between, how well can you tell the difference between one and then the other. So that's called sequential. They're delivered in sequence. Then we get a number for that. Then we deliver both at the exact same time. When you deliver at the same time, now you're really tasking this inhibition. When you're doing it sequentially, you have no trouble telling which one's larger. When you do them both at the same time, now you're starting to have a little bit of trouble, especially if you had a few beers, or if you are asleep, or if there's something wrong, you have a lot of trouble. And so the important measure to look at is, how does your simultaneous amplitude discrimination compare to your sequential amplitude discrimination? That's all part of your accuracy score. How well can you refine those measures, and how well can you tell the difference?
I'd like to make a comment here, because this is really what our companies are about. This is what our academic research is about. This is what we're trying to do. So this is where engineering and life science, brain science comes together. And so this is, there's brain science in here. This tells you something about how the brain works. And to get at how the brain is working, we use the engineering tricks of, signal reception and signal processing to analyze this data. And we use basic approaches to engineering to get a good signal. And this is like measuring a difference. So we're measuring this differential signal, and the way that it's measured in the brain gauge allows us to cancel out most of the things that are going on in your periphery, in your hands and in your peripheral nerves and so forth. And these things, if they're roughly the same value, they'll tend to cancel out. And we can focus in on what's happening in the brain by testing how you perceive the differences between two fingertips that are adjacent spots on your somatosensory cortex. So this is, what's needed here. What's been needed for decades is an intelligent combination of engineering, practices and engineering techniques with biology, focused on telling you something useful. And that's what brain gauge is all about. That's what we do. So
one last comment about this. A lot of people have asked us over the last few weeks, how does this compare to MRI, fMRI, functional imaging, or taking a scan to see how my brain does? And for the record, most MRIs show nothing for something like concussion. But one thing is, if you do a functional MRI and you deliver one stimulus to one finger and take a picture, take an image, and then you deliver another stimulus to another finger, you might be able to see where those two areas of activation are, but you won't be able to see the 10% differential that you can see with sensory percept. So people over the last few years, last few decades, have really become to rely on technology to tell them how good things are. And it basically would be a little bit like going to get your eyeglass prescription and getting put in a scanner. I don't think anybody's ever done that, because eye charts work really well. That is a sensory perceptual measure. And the thing to remember is that measures of sensory percept are the highest resolution measure you can get in terms of looking at these subtle differences in variation.
Now, this is where I disagree with Dr. Tommerdahl, if I may. Full deference to my esteemed colleague.
All right, didn't think we ever disagreed.
We rarely disagree, but this is one of these points. Now, he says you can't see anything when you go to get an MRI, and in fact, I know you can see something. if you bump your head and you go and get an MRI, you'll see one thing for sure. You'll see an extremely large bill, and your insurance may or may not pay for it. What does that tell you about your brain function? I don't know. But there's a lot of things you can pay a lot of money for and learn nothing of value about your health status. So we're trying to do just the opposite and provide tools that don't cost a lot of money. They cost something, but it costs us millions of dollars.
Yeah.
I don't even want to take a guess what we spent on developing the brain gauge, but it's been a lot of money every year for 10 or 12 years. So
the downside of the brain gauge for measures like this, or any of the measures actually, is how sensitive it is. It's actually extremely sensitive to a lot of different things.
That's the problem.
So if you're trying to look at, say, well, how do I change from one year to the next, and you measure yourself 1 year and the next year, but 1 year when you measure, well, you drank all weekend and then measured, you might not do too well, and the next year you might, not gotten any sleep and still didn't do too well, or maybe you were optimum one time and then not optimum another. You need to do a little bit of work to figure out just what your brain function is.
And the thing is, it's sort of like you can test your eyesight, which will be kind of different at different times of the day. I don't know if you've noticed, it's harder to focus when you're tired and stuff like that. And if you test your blood pressure, it's different. You test your body temperature, any vital sign changes through the day somewhat. So, you can weigh yourself, and you're going to weigh a different amount, morning versus night on average. So the brain gauge needs to be looked at. The measures that you get need to be looked at that very same way. They were very, very precise. They're very, very repeatable. And we think they tell you something very important about, how your brain's functioning. But you got to be kind of intelligent about it, and you realize that you do have diurnal, day throughout the day variability, day to day and week upon week. It's something where you kind of want to look at trends, then you want to see it in the greater, the greater context, right? So one of the things that Mark hasn't talked about is that he's got how many people in your database now? 10,000?
It's about 10,000 plus.
Yeah, so it's a lot of people. Every time I ask, it's like 1,000 more people. So he can, the brain gauge is designed to give you insight into where your function is within that group of people. So, there's a lot of information embedded in it, but once again, you don't want to take one measure and put too much stock in one thing. You don't want to look at one change and put too much stock in it. But if you use it intelligently, over time, it tells you quite a lot.
Yep.
Anything else?
I think that's our shortest video
yet. And I'm out of beer, so those are the rules. And we'll see you again next time.
Alrighty. Bye-bye.